Chapter 19. Animals. The ultimate design of life. A cell without a cell wall is inherently fragile, struggling to withstand the stringencies of nature and easily becoming prey. To counter this vulnerability, such cells often cluster together, forming clumps that enhance their stability and chances of survival. While multicellularity is observed in plants and fungi, which possess protective cell walls, it is an option for them rather than a necessity. However, for organisms without cell walls, multicellularity becomes an absolute obligation. Animals consume food, break it down through digestion, and extract matter and energy from it. Waste products from metabolism, such as feces and urine, are excreted. Unlike bacteria, fungi, and plants, animal cells do not have cell walls, which presents a challenge in maintaining shape. The absence of a cell wall can compromise the cell's rigidity, making it difficult to form a stable structure. However, this lack of a cell wall allows for important aspects such as nutrient uptake and mobility. To overcome this challenge, animals have evolved skeletal structures. Insects have exoskeletons, while most other animals have endoskeletons. The body's integrity is maintained through numerous interactions between cells and an extensive extracellular matrix, which shapes cells, tissues, and organs and provides a degree of flexibility and softness. The absence of cell walls in animals is compensated for by the skin, protection, intercellular interactions, cellular and tissue shape, extracellular matrix, tissue and organ shape, and skeletal structures, organismal shape. Notably, the lack of a cell wall allows for extensive cell-to-cell interactions and the potential for dynamic shape changes. This is a key factor in the emergence of movement in multicellular animals. Multicellularity can be broadly classified into two types based on the presence or absence of cell walls. Plants and fungi, characterized by their rigid cell walls, represent one category of multicellular organisms. Animals, on the other hand, fall into the category of multicellularity without cell walls. This distinction significantly impacts the degree of dynamism observed within these organisms. Plants and fungi, constrained by their cell walls, exhibit relatively less dynamic development. In contrast, animals, unbound by the rigidity of cell walls, showcase extraordinary dynamism throughout their developmental stages, from the zygote to the fully developed organism. The absence of cell walls allows animals to undergo complex cellular movements, forming and reforming diverse structures, embodying an exceptional level of developmental fluidity and adaptability. In animals, the journey from a single-celled zygote to a fully formed infant is a captivating display of cellular choreography akin to a drama or ballet. Cells move, form, and reform diverse structures as the organism progresses through various developmental stages. This intricate process is orchestrated by morphogens, specialized signaling molecules that guide cellular differentiation and tissue formation with remarkable precision. An equally critical aspect of animal development is programmed cell death, apoptosis, which allows the removal or regression of organs or tissues that serve a temporary purpose during development but are no longer required, ensuring the organism's optimal structure and function. Animal multicellularity introduces significant challenges, particularly due to the need for aerobic respiration and the acquisition of food from external sources, heterotrophy. To maintain life, animals must efficiently deliver oxygen and nutrients to every cell. This requires complex systems for food acquisition, digestion, nutrient assimilation, waste elimination, and circulation. The circulatory system plays a vital role in distributing essential resources throughout the body. To coordinate these processes, animals have evolved a nervous system that enables them to respond to both internal and external stimuli, ensuring that all organs work together to support the organism's survival. Metabolism. The diversity of metabolism is as vast as the organisms themselves. Bacteria, fungi, and plants obtain nutrients through amino acids, simple carbohydrates, and similar small organic compounds. Generally, their cells do not absorb complex or large molecules. For instance, bacteria possess a cell membrane covered with a peptide-glycan cell wall. Most large molecules, including proteins, pass through the cell wall and are broken down into amino acids, which are then absorbed through the cell membrane. This process of digestion occurs within the periplasmic space, which is the area between the cell wall and cell membrane. Fungi, which have cell walls made of chitin, secrete digestive enzymes that break down dead materials. The simpler molecules are then absorbed by root-like structures and transported throughout the rest of the mushroom. Plants, with cell walls made of cellulose, absorb simple molecules from the soil, which are released by microorganisms or other soil-dwelling organisms. They use solar energy to synthesize the necessary biomolecules. Protists, such as amoebas, which lack a cell wall, and animals, which also lack a cell wall, acquire nutrients within their organisms. Complex food particles enter their bodies, and undigested particles are expelled. All animals are heterotrophs, meaning they consume other living matter. They assimilate the matter and energy derived from their food into their bodies. Our bodies have an alimentary canal where food is digested into smaller molecules like glucose and amino acids. These molecules are then transported throughout the body via the bloodstream. Undigested matter is excreted as feces. All organisms assimilate simple molecules obtained from their environment. Some protists and phagocytes of the immune system absorb large complex molecules, such as bacteria, and digest them within their cells. Organisms can then use the simpler molecules derived from digestion to obtain energy and build materials. The process of digestion and absorption of food differs between organisms with and without a cell wall, particularly in terms of how nutrition crosses the cell membrane. In organisms with cell walls, digestion typically occurs outside the cell membrane. In contrast, digestion can occur within the cell or within the organism in organisms without cell walls. If an organism consumes easily usable materials, there is no issue of undigested or unassimilated food. It is always preferable for the cell not to have to deal with waste material, which should be excreted. This is especially important for multicellular organisms. There should be a mechanism to collect all waste materials from all cells and excrete them. However, evidence of active excretion is scarce in most multicellular organisms with cell walls. In animal cells, a whole organ system is dedicated to digestion, absorption of nutrients, and excretion of undigested material. Furthermore, liquid tissue systems circulate essential substances such as oxygen and nutrients while flushing out waste materials. In animals, growth involves increasing the size of all body parts. In plants, growth mainly occurs at the tips and surfaces. In animals, the size of most organs increases from birth until adulthood. The skin, heart, brain, and skeleton are smaller at birth, but grow in size over time until adulthood. This means that the cells of different organs continue to divide while old cells die and are often recycled. For example, old skin cells, hair, and nails die and contribute to the outer layers of these protective structures. Animal body organization is dynamic, with cells wearing out and organs being replenished with new cells. Some organs, like the heart and brain, are believed to remain constant, post-mitotic organs, after reaching full size, but this is still debatable. The genome is the most stable molecule, without much of a turnover within a cell. All other molecules and structures undergo turnover. Homeotherms are organisms that maintain a relatively constant body temperature regardless of external environmental conditions. They achieve this through physiological and behavioral mechanisms such as regulating metabolic rate, insulation, and thermoregulatory behaviors. On the other hand, poikilotherms are organisms whose body temperature fluctuates with the temperature of their environment. Variations influence their metabolic rate and activity levels in external temperature. Poikilotherms often rely on behavioral adaptations, such as basking in the sun or seeking shade, to regulate their body temperature. While homeotherms can maintain a stable internal environment, poikilotherms exhibit greater flexibility in their energy requirements. They can survive in a wider range of environments. Growth, differentiation, and development. Animals start as a zygote, a single cell formed from the fusion of sperm and egg. Our entire body originates from this single cell. The zygote continuously divides and forms a ball of identical cells called a morula. The morula then develops into a blastocyst, with a gap in the center resembling a football. Within the blastocyst are two cell types: the inner cell mass, a clump of cells, and the blastomeres, the outer single cell layer. This is when differentiation occurs, with multiple cell types forming despite having the same genotype. All the tissues in our body come from the inner cell mass, the source of embryonic stem cells. Our body consists of about 210 different cell types, all with the same genetic material, forming the somatoplasm. Somatoplasm refers to the cellular material that forms the body's somatic cells, which contribute to tissues and organs. These cells arise through the differentiation of the three primary germ layers. 1. Ectoderm forms the skin and nervous system. 2. Mesoderm gives rise to muscle, bone, blood, and internal structures like the heart. 3. Endoderm develops into organs like the lungs, liver, and parts of the digestive system. Germplasm, conversely, consists of primordial germ cells, PGCs, which give rise to gametes, sperm and eggs. This ensures the continuation of genetic material to the next generation. Of the body, germplasm undergoes the least number of replications. Cell number homeostasis refers to maintaining a stable number of cells in a tissue or organ, which is essential for proper functioning and survival. It is regulated through a feedback loop that balances cell production, proliferation, and cell death, apoptosis. When differentiated cells undergo wear and tear or damage, the drop in function signals triggers increased cell production to replace lost cells. Adult stem cells play a critical role by dividing into new cells that differentiate into functional cell types. This ensures tissues maintain an optimal number of cells for proper body functions. If cell death rates surpass production, tissues experience degeneration, leading to conditions like aging or disease. Conversely, uncontrolled cell proliferation, such as tumors, can result in abnormal growth. The feedback loop works by signaling mechanisms involving growth factors, cytokines, and internal cellular checkpoints. For example, when tissue damage or cell loss occurs, signals stimulate stem cells to divide, increasing the production rate. Once the optimal cell number is restored, inhibitory signals reduce further proliferation. This dynamic balance ensures tissue integrity and functionality. Disruption in this balance, such as reduced apoptosis or excessive proliferation, can lead to diseases like cancer or tissue atrophy, underscoring the importance of feedback regulation in cell number homeostasis. Cells of the same type interact through cell surface protein-protein interactions and can also interact with nearby cell types. Additionally, all cell types are connected through the circulatory system. Glands produce signals in the form of hormones circulated throughout the body and received by target cells. The functional differentiation of cells is dictated by expressed genes, physical interactions with surrounding cells, and signals from different locations in the body. Different organs carry out specific functions: lungs for gaseous exchange, heart for pumping blood, blood for circulation of oxygen and nutrients, alimentary canal for digestion, kidneys for excretion, and skin for protection and temperature regulation. The endocrine system regulates growth, development, and metabolism, while the nervous system signals bodily functions, perceives stimuli, and responds to them. Each organ has its own adult stem cells derived from primordial germ layers. These adult stem cells continuously produce new cells to increase size or replace worn-out cells. Each organ maintains an optimal number of cells for optimal functioning based on age and body size. The absence of cell walls in animals has allowed for flexibility and complexity in their development, making them the superstars of evolution. Early embryogenesis in humans is a highly intricate and regulated process influenced by genetic and environmental factors. Any disruptions or abnormalities during this critical period can have significant consequences in subsequent development, potentially leading to congenital malformations or developmental disorders. Maintenance, repair, and regeneration. The maintenance and repair of the body rely on adult stem cells, which ensure tissue regeneration and homeostasis. Adult stem cells have the unique ability to self-renew and differentiate into specialized cell types. They are key in maintaining tissues, particularly those with high turnover rates like skin, blood, and the intestinal lining. The diagram illustrates two primary processes: self-renewal and asymmetric cell division. In self-renewal, stem cells replicate to maintain their population, without which the organs can no longer regenerate or repair. In asymmetric division, One daughter cell becomes a transit amplifying cell, TAC, while the other retains its stem cell properties. TACs undergo limited rounds of rapid division before committing to a specific lineage and differentiating into functional, terminally differentiated cells. These cells perform specialized roles such as providing structural support or metabolic function. Tissue maintenance is essential for replacing damaged, old, or dead cells. In tissues like skin or blood, stem cells are constantly active. However, stem cells are quiescent and activate only during injury in organs with slower regeneration, like nerves or muscles. A precise balance between self-renewal and differentiation is crucial. Imbalances may lead to stem cell exhaustion, aging, or uncontrolled proliferation, resulting in diseases like cancer. Because animals are heterotrophs, they are always susceptible to different pathogens. Additionally, animals often have to engage in strenuous activities to hunt or avoid being hunted, putting them at risk of injuries and accidents. As a result, animals require complex systems to defend their bodies against infections and to aid in their recovery after accidents. The increased risk of injury also heightens the chances of infections occurring. The immune system has evolved in the animal kingdom to survive these challenges. The immune system provides immediate nonspecific protection as well as long-term specific protection. The nonspecific immune system functions by eliminating anything that is perceived as foreign. Nonspecific immune cells typically safeguard body parts that are particularly prone to infection or serve as entry points for pathogens. Orifices such as the eyes, nose, mouth, genitals, and anus are constantly protected by immune cells and their components. If a pathogen enters the body, the immune system employs 3 strategies to eliminate it: degradation, eviction, and encapsulation. These strategies aim to eradicate foreign objects. The immune system often accomplishes this by breaking down the pathogen or foreign body. Pathogens may be engulfed by immune cells or broken down by antibodies in conjunction with the complement system, another set of immune system components. Regeneration is a fascinating phenomenon observed in various animal species across the animal kingdom. It refers to the ability of an organism to regrow or replace damaged or lost body parts, ranging from limbs and organs to tissues and cells. While not all animals possess the same regenerative capabilities, many exhibit remarkable regenerative abilities that allow them to recover from injuries in extraordinary ways. One of the most well-known examples of regeneration is found in starfish. If a starfish loses an arm, it can regrow the missing arm through a process called epimorphosis. Specialized cells at the site of the injury dedifferentiate, transforming into a mass of stem-like cells known as blastema. These cells then divide and differentiate to form the new arm, eventually growing into a fully functional structure. Planarians, flatworms known for their incredible regenerative capabilities, can regenerate complete organisms from tiny fragments. Even when cut into several pieces, each fragment has the potential to regenerate into a new worm, including all necessary organs and tissues. This is possible due to the presence of pluripotent stem cells throughout their bodies. In the case of salamanders and newts, they can regenerate entire limbs, spinal cords, and even parts of their hearts. When a limb is lost, a complex series of events is triggered, involving the formation of a specialized structure called the blastema. The blastema consists of undifferentiated cells that proliferate and differentiate into various cell types required for the formation of a new limb. Certain lizards, such as the green anole, can regenerate their tails. When a tail is autotomized, self-amputated, the lizard's immune system and wound healing processes are activated. The regeneration process involves the formation of a blastema, which then differentiates into a new spinal cord, muscle, blood vessels, and skin, resulting in a complete and functional tail. While humans possess some regenerative capabilities, such as the regeneration of liver tissue, our regenerative abilities are limited compared to these remarkable examples. Studying the regenerative processes in animals may provide insights into potential therapeutic strategies for human medicine, aiming to enhance tissue repair and regeneration in the future. The nervous system, sensory organs, motility, and instincts. Organisms with differentiated cells have different characteristics, leading to variations in behavior. However, if organs composed of different cell types work independently, the entire system will collapse. Thus, coordination among organs is crucial for the survival and reproduction of organisms. To achieve this, organisms have evolved specialized cells called neurons, which integrate all the organs into a cohesive unit. Neurons are elongated and can form chains, allowing them to communicate signals throughout the body. Throughout evolution, the nervous system in animals has become intricate and diverse. It originated in simple multicellular organisms and gradually developed into complex networks in more advanced organisms. Primitive organisms like jellyfish had decentralized nerve nets made of interconnected neurons. In contrast, worms and arthropods developed ganglia, enabling coordinated movement and responses to stimuli. A significant milestone was the evolution of a central nervous system, CNS, observed in mollusks and vertebrates. The CNS, consisting of the brain and spinal cord, processes and integrates sensory information, enabling more sophisticated responses. This laid the foundation for complex behaviors, learning, and memory. Memory is the basis for all learning, and memory of past experiences shapes the organism's future behavior. The memory of past pains and pleasures is the key to the organism's existence. The memory of pain keeps us from injury. In contrast, the memory of pleasure drives us to find more, usually food, reproduction, and social connection. The emergence of memory and movement is the key to the flourishing existence of animals. Animals have a purposeful action to fulfill the biological objectives of existence and reproduction. The brain underwent further evolution in vertebrates, with distinct regions responsible for sensory processing, motor control, and higher cognitive processes. In mammals, particularly, the cerebral cortex expanded significantly in size and complexity, contributing to advanced cognitive abilities. Specialized sensory organs, such as eyes and ears, also emerged, enhancing the detection and interpretation of environmental stimuli. The evolution of the nervous system is closely linked to its adaptive advantage in perceiving and responding to the environment. The diverse and complex neural structures in different animal species reflect the varied ecological niches and behavioral strategies they have developed to thrive. Humans have the most complex brains. Therefore, I opine that humans are the pinnacle of evolution and emergence. Nothing to be modest about. As organismal complexity increases, neurons organize into ganglia, which serve as information processing and decision-making centers. In higher organisms, complex brains have evolved to control the body. Brains are usually located near sense organs and allocate a significant portion of their capacity to sensing information from the external world. Subsequently, the brain instructs the body's movements. The nervous system operates through input, processing, and output. Physical stimuli such as sound, smell, taste, emotion, light, temperature, and touch serve as the input which is perceived within the brain. Processing is a complex process that may involve analyzing past experiences. In higher organisms, the nervous system has become more complex, allowing for the ability to memorize, learn from experience, communicate, and predict. The nervous system coordinates the entire body and its interactions with the environment. Different animals have specialized organs to sense chemicals, vibrations, pressure, temperature, sounds, and light in their surroundings. The nose detects chemicals in the air, smell. The tongue detects chemicals in food, taste. The eyes detect light, vision. The ears detect sound, hearing. And the skin detects temperature and touch. The cells responsible for sensing and executing actions are specialized cells. All communication between these cells occurs through the nervous system. Sensory organs receive information and send it to the brain, which perceives and responds based on context and need. The brain then relays information to the muscles, which create appropriate actions. Animals can also produce signals, such as chemical, electrical, vibrational, auditory, and visual signals. Pheromones and body smell serve as signals between individuals. Fireflies emit light as a mating signal, and animals and humans use sound for communication. If all the cells in an organism had rigid cell walls, the organism would struggle to position body parts in relation to each other. For body parts to move independently, a support system is necessary. Animals have hard tissues, such as the skeletal system, which are highly dense with mineral deposits and largely inactive in terms of metabolism. Exoskeletons and endoskeletons provide shape and protection to organisms. Endoskeletons, like those in vertebrates, consist of multiple bones joined together by ligaments. The motility of an animal relies on the bones acting as livers, ligaments holding bones together in joints, tendons attaching muscles to bones, and muscles generating mechanical energy. The nervous system regulates the functions of all organs in a coordinated manner. Most animals have a nervous system that communicates through electrical impulses and forms networks of neurons, enabling highly complex functions such as learning, memorizing, thinking, and predicting. The nervous system enables coordination, agility, planning, and decision-making in animals, including during fights between two tigers. Similarly, in games like badminton or tennis, players need agility, strength, coordination, and the ability to read opponents and teammates, respond accurately, and control inanimate objects with other inanimate objects. The nervous system essentially orchestrates these activities. Muscle movement in animals is facilitated by the coordinated action of the nervous system and the absence of a cell wall. The nervous system transmits electrical signals from the brain and spinal cord to the muscles, enabling them to contract and generate movement. Nerve impulses travel along specialized cells called neurons, allowing precise control over muscle contractions. Unlike plant cells, animal cells lack a rigid cell wall, which provides flexibility and allows for the contraction and relaxation of muscles. This flexibility enables animals to exhibit various movements from simple reflexes to complex behaviors. Motility enhances the survival game. It enables animals to chase, run, fight, and kill. It allows them to move towards sources of food and away from predators. The presence of motility dramatically increases the number of dynamic interactions and creates unusual scenarios. Motility provides the freedom to find food and avoid harsh conditions. When both predator and prey are motile, fierce chases become a daily sport in nature. Physical fitness, in the form of speed, agility, and endurance, becomes crucial. It also drives the formation of groups for hunting and escaping. Thanks to motility, organisms have transitioned from the sea to the land, from the land to the trees, and to the skies. Animals with motility can occupy all available spaces on the planet. For example, a puffin bird can walk on land, fly in the sky, and swim in water. The animal kingdom has invaded almost every available resource with specialized adaptations. Motility also allows for engineering structures such as nests, webs, and even cars. The ability to engineer structures provides a fitness advantage, as animals can manipulate their environments to evade environmental stress. Considering the functional convergence and divergence of organs in different species is interesting in animals. To illustrate functional divergence, let us discuss the various functions of tails. Most animals have unbranched tails that extend from the rear of their bodies. However, different species utilize tails for different purposes. In fish, tails function as rudders, enabling them to swim. In birds, tails help regulate flight. In certain reptiles, tails serve as a formidable defense against predators. In mammals, tails have evolved to fulfill a wide range of functions that contribute to their survival and adaptation. One important role of tails is to maintain balance and stability during movement. Animals like cats, monkeys, and kangaroos use their tails to adjust their body position and remain steady while navigating various terrains. Tails also play a crucial role in communication among mammals. They serve to convey signals, emotions, and social cues to others of the same species. For example, a wagging tail in dogs signifies excitement or friendliness, while a tucked tail indicates fear or submission. Some mammals have prehensile tails, which have adapted to grip and hold objects. Monkeys and opossums, for instance, utilize their prehensile tails as an additional limb to grasp branches or objects, aiding in locomotion and foraging. Tails can also serve as a means of defense and protection against predators. Certain mammals, like porcupines and scorpions, possess tails armed with defensive structures such as quills or stingers, which can be used as weapons. Additionally, tails act as a site for fat storage in specific mammals, serving as an energy reserve during times of scarcity. Kangaroos and fat-tailed lemurs are notable examples of tails also for fat storage. For aquatic mammals like whales, dolphins, and seals, the tail acts as a powerful propeller for swimming, enabling efficient movement through water. The functions of tails in mammals are diverse and vary depending on the species and its ecological niche. Physical and mental plasticity. Physical and mental plasticity in humans refers to our brains and body's capacity to adapt and change throughout our lives. These processes are essential for acquiring new skills, improving cognitive functions, and recovering from injuries or challenges. Physical plasticity, neuroplasticity, the brain's ability to reorganize itself allows us to learn, remember, and adapt to new information and experiences. This plasticity enables us to recover after brain injuries and acquire new skills. Muscle memory. Our muscles and motor systems also exhibit plasticity. With consistent practice, we can improve physical abilities such as playing musical instruments, participating in sports, or developing fine motor skills. Aging and exercise. Regular physical activity can mitigate the decline in muscle and bone mass that occurs with age, promoting physical plasticity. Exercise also supports cognitive function by enhancing blood flow and neurogenesis in the brain. Mental plasticity, learning and memory. Our ability to acquire knowledge and retain information relies on mental plasticity. Learning new languages, mastering complex subjects, and adapting to changing technologies all rely on our brain's ability to rewire itself. Emotional regulation. Emotional plasticity refers to our ability to adapt our emotional responses to different situations. Through therapy, mindfulness practices, or self-help techniques, we can modify emotional reactions and build emotional resilience. Creativity and innovation. Mental plasticity is at the core of human creativity. The brain can forge new connections between seemingly unrelated ideas and concepts, fostering innovation in art, science, and technology. Resilience and adaptation. Humans exhibit mental plasticity when facing adversity or trauma. With appropriate support and coping strategies, individuals can recover from setbacks, develop resilience, and adapt to new circumstances. Cognitive training. Exercises like puzzles and brain games can enhance mental plasticity by challenging and strengthening cognitive functions like problem solving, memory, and attention. Lifelong learning. Lifelong learning highlights our innate mental plasticity. Throughout our lives, we can continually acquire new knowledge and skills, keeping our minds active and adaptable. Physical and mental plasticity in humans is vital for our growth, development, and overall well-being. These processes underlie our ability to learn, adapt, and thrive in an ever-changing world, making us versatile and resilient beings. Parental care. Parental care is a crucial aspect of animal reproductive strategies. It involves parents' dedication of time, energy, and resources to nurture and protect their offspring. The animal kingdom demonstrates a wide range of parental care behaviors, including maternal care, paternal care, biparental care, cooperative care, and brood parasitism. Maternal care encompasses activities such as nest building, incubating eggs, and nursing or feeding the young, and mothers primarily provide it. Mammals, birds, and certain reptiles, such as crocodiles, showcase extensive maternal care. In some species, fathers also play a significant role in offspring care, known as paternal care. They participate in tasks such as guarding nests, protecting young from predators, and providing food. Seahorses are a notable example of paternal care, with males carrying and incubating eggs. Biparental care involves both parents sharing responsibilities in raising their offspring. Both parents share tasks such as building nests, feeding, and protecting the young. Bird species like pigeons and penguins display biparental care, taking turns in incubating eggs and feeding the chicks. Cooperative care involves assistance from multiple individuals, often non-parents, in caring for the offspring. Social insects like ants and bees exemplify cooperative care, with workers helping feed, clean, and protect the young. In contrast, brood parasitism is a unique strategy where certain species lay their eggs in the nests of others, leaving the host parents to care for and raise their young. The common cuckoo is a well-known example of a brood parasite. The type and extent of parental care vary greatly across species and are influenced by factors such as environmental conditions, available resources, and social structures. Parental care ensures the survival, growth, and successful transition to independence of offspring, contributing to their overall well-being in the animal kingdom. In the animal kingdom, certain species exhibit behaviors where parents abandon or allow the death of weaker offspring, often referred to as the runt. For instance, gray wolves may let the weakest pups die to ensure that stronger ones receive more resources. Kangaroos and cheetahs sometimes abandon the runt if resources are scarce, focusing their energy on the most viable offspring. Lions may reject weaker cubs, particularly when food is limited or when new males take over the pride. This strategy, seen across various species, ensures that limited resources are directed toward the offspring with the best chances of survival. Aging: all animals age and eventually die. Aging is a natural process that occurs as animals get older, resulting in a gradual decline in physiological functions and an increased susceptibility to age-related diseases. At the cellular level, aging involves a decrease in the ability of cells to divide and repair themselves, leading to accumulated damage to cellular components. This damage can impair cellular function and cause tissue dysfunction. As animals age, the functioning of their organs and systems can decline. For example, the cardiovascular system may become less efficient, leading to reduced blood flow and impaired oxygen delivery. The immune system may weaken, increasing the risk of infections and diseases. Senescence, which is the loss of cell division ability and irreversible growth arrest, is a characteristic of aging. Senescent cells can accumulate in tissues over time, contributing to age-related conditions and inflammation. Aging is a significant risk factor for various diseases in animals, including neurodegenerative disorders, cancer, arthritis, and cardiovascular diseases. The cumulative effects of aging on cells and tissues make animals more vulnerable to these conditions. Aging is a multifaceted process influenced by several key factors. First, the accumulation of mutations over time can damage DNA, leading to cellular dysfunction and aging-related diseases. Second, the shortening of telomeres, the protective caps on chromosomes, occurs with each cell division, eventually leading to cell senescence and a decline in tissue regeneration. Finally, the exhaustion of adult stem cells, which are responsible for tissue repair and regeneration, limits the body's ability to maintain and restore damaged tissues. Together, these processes accelerate the decline in cellular function and organismal vitality, contributing to aging. Different animal species exhibit variation in lifespan, which is influenced by factors such as genetics, environment, and lifestyle. Some animals demonstrate exceptional longevity compared to others. While aging is a universal process, its rate and manifestation can vary among species. The varying lifespans among animals within the same order can be attributed to a complex interplay of genetic, ecological, and evolutionary factors. The classification of organisms at the order level in biology encompasses those that share certain fundamental characteristics. However, within these groups, species exhibit remarkable diversity in their life expectancies. A prime example can be found among primates. Some members of the order Primates, such as the mouse lemur, Microcebus murinus, have relatively short lifespans of around 6 to 8 years, while others, like humans, Homo sapiens, can live well beyond 70 years. One crucial determinant of lifespan variation is the pace of life strategy. Species with a fast-paced life history, characterized by rapid reproduction, early maturation, and higher mortality rates, tend to have shorter lifespans. In contrast, those with a slower pace of life, investing more in fewer offspring, and exhibiting delayed maturation, often enjoy extended lifespans. For instance, the African elephant, Loxodonta africana, belonging to the order Proboscidea, is known for its relatively long lifespan, living up to 60 to 70 years. Ecological niches and environmental pressures also influence the variation in lifespan within the same order. In birds, for example, members of the order Passeriformes, like the common house sparrow, Passer domesticus, may have a lifespan of around 3 to 4 years, while larger species like the albatross, Diomedia excellens, can live for several decades. Evolutionary pressures, including predation risks, environmental stability, and reproductive strategies, further contribute to divergent lifespans. Species facing high predation pressure may exhibit shorter lifespans as a survival strategy, investing more in rapid reproduction to counteract high mortality rates. On the other hand, species in more stable environments with fewer predators may evolve longer lifespans, emphasizing the importance of reproductive strategies tailored to specific ecological contexts. The order Carnivora offers examples such as the comparatively short-lived domestic cat, Felis catus, and the longer-lived African lion, Panthera leo. The factors influencing lifespan within the same order are multifaceted, encompassing pace of life strategies, ecological niches, evolutionary pressures, and reproductive tactics. The variations observed underscore the complexity of life history evolution, highlighting the diverse pathways organisms within the same order may take to adapt and thrive in their respective environments.